So our first keynote speaker is Dr. Sander Bohte from CWI Machine Learning Group. He's a senior researcher in that group and also a part-time full professor of the Cognitive Computational Neuroscience at the University of Amsterdam and a part-time full professor of bio-inspired neural networks at the University of Groningen in the Netherlands. He received his PhD in 2003 at CWI on the topic of spiking neural networks. His work has been pioneering in the development of advanced and efficient spiking neural networks, including seminal work on supervised learning with spike time-coded networks. Recent work has also developed biologically plausible deep learning and deep reinforcement learning models for cognition and spiking neural network versions thereof. Sander, thank you so much for speaking to us today, and I'll turn it over to you.
Yes, thank you, Katie. So first of all, thank you very much for the invitation. And also thank you very much for all the hard work that went into this conference. I realize how hard it must have been to change everything. Yes, so computing with spiking neurons. I've been doing it for a while. So here's the quick outline of the talk for today. I thought I'd discuss why spikes. I've been doing it for, I guess, 23 years now. Why spikes? It's getting a little bit easier, but it might be good to revisit that, especially now that it's getting easier. And I also want to talk a little bit about real biological neurons and some of the things that are really cool and interesting about real neurons that we're not really using in spiking neural networks at this point, or we're starting to use. And then I can actually give one of the examples where we are using these features from real spiking neurons, and we find that it's actually very useful to get good results. And I'm not quite sure whether I'm going to get to the reinforcement learning for cognition. I hope to touch a little bit on biological plausible deep learning and how that relates to spiking neurons. I do want to touch a little bit on open questions and challenges as I see them, as we've been discussing them over the last few months in all various kind of projects. That's what you do when you're stuck at home. You write projects and papers, fortunately, but mostly projects. Yeah, so first of all, so why did I get into spiking neurons way back in '97? So as a student, I was convinced that with ever growing computers, that AI was going to be the big thing, and it was going to be built on neural networks. And if you go way back to '97 when this was happening, that was exceptionally poor timing, as you can see on the slides, and because clearly standard neural networks were not working. Even though Schmid-Huber and Hochreiter came out with LSTM in '97, it took a while before that was picked up. And at that point, I think in '97, even the NeurIPS conference, which is Neural Information Processing System, didn't have any papers on neural networks. So that's just to go back in memory lane a little bit. And then there was this paper from the professor Wolfgang Maas from Austria, and he was describing this new generation of neural networks, spiking neural networks, and that might just be what you need to get AI going with neural networks. So this was actually '97. I was finishing up my master thesis with spiking neurons in computational neuroscience, and I wanted to go do that. And of course, in the back of your mind, you also have this idea that if you're ever going to merge AI in some way with actual humans, you're going to have to talk the language of the brain, which is spiking neurons. That's a way back, but I think it's much more relevant now. So even though it's been another winter, I think I've used some of that winter fairly well. So I know a lot about actual biology. I'm even part-time professor now in the biology department. That's rather surprising. So I hope to give you some of that information of what I learned, and it might be useful here. But first off, the standard slide deck of why spiking neurons. So these are the arguments that we now kind of give on an almost standard basis, which says, well, first of all, your brain is huge, lots of neurons, in a humongous amount of connections. But these neurons, they don't do very much. They exchange these spikes once in a while, 1 to 5 hertz on average. And they run at like 25 watts. And 25 watts is not a lot of energy. It's actually a small light bulb for those who remember. And it's massively more efficient than even a huge supercomputer that might or might not get into the same neighborhood of computational power. That's also a big part of the debate. So that's now a big reason for looking at spiking neural networks. Can we get this efficiency per neuron out of the brain? Can we get that into algorithms? Can we get it into hardware? Specifically, of course, with edge AI applications in mind, things that live on a chip, on a battery, that don't need to connect to the cloud, but still needs to do something useful. So that's a big one. I think that's been recognized quite well. Here we are back to that connection to the brain, or to the nervous system in this case. As I said, the brain works with spikes and muscles are controlled with spikes. So if you are looking into neuroprosthetics, and people are increasingly doing that, and I have some very cool results that are coming out, including groups here around in Amsterdam, actually. You are going to talk spikes, and you need to know real spikes, or real spiking neurons. So finally, and this is also a topic that is starting to emerge, I think. It's an argument from robotics. And it says, we are building these very complicated robots now, and they have tons of sensors that each run at their own sampling rate. And we also have a whole bunch of actuators, and they also need to be controlled. And if you do that in a synchronized manner, as you would do with a CPU, you have a tremendous coordination problem. You need to get information from all these sensors. You need to somehow sort them out and put them together and then control these outputs. And what you really want to do is some kind of asynchronous processing in an event-based manner. And that's, of course, exactly what spiking neural networks do. So this is sort of the third big application domain that may not be fully developed, but I think a lot of people are working on it, not me. And then, of course, there's these things coming up now to show that this field is really taking off. There are Nature papers, which is about as far as you can get, of course, on neuromorphics. And there are real neuromorphic chips, no longer toy examples or tiny things. You cannot quite buy them, but I think if you send an email to Intel, you can get access. We do, and I think it was pretty easy after some paperwork. So there's actual real chips to work with, real spiking neural network chips. So that's very, very cool. Of course, then there are people like this. Some more convincing is needed in places. And here the main argument that Jan LeCun was making was that all these spike-based neural networks are not all that good. And of course, we can argue about that. But to some degree, I think at that point, at least, he was somewhat right. At best, things came a little bit close, but were very expensive. There was no advantage. And if you don't have good algorithms, why would you build chips? And of course, Mike Davies will say then, well, if you don't build chips, there will be no algorithms. So you need to start somewhere, and I think he's right on that. But anyway, this is the challenge that's been placed by Jan LeCun. I have to give you some examples of how far we are getting now. So first, to go back to biology, I hope to teach you a little bit of biology. So neurons in the brain, how many neurons are there actually? And it's kind of cool to see that even insects, they don't have that many neurons. They have a couple thousand for like an ant, which is really next to nothing. Or a bee has 1 million neurons. And a bee, you can already teach tricks. That's kind of cool. You can teach it to choose between different smells. And of course, we know that bees can communicate with each other. So even with 1 million neurons, you can exchange navigation cues. Or at least that's how we think it's been done. Now then at the other end, before we get to humans, which have huge brains, and it's going to be a while even before Henry Markram is going to put that brain on a big supercomputer. But a simple rat has about 100 million neurons. And that's kind of cool because a rat is a fantastic robot, of course. It's very agile, very well coordinated. It also has pretty good sensory processing. It may not see very well because its eyes are not that good, but it's very good at sensing and smelling. And of course, you can also teach it tricks. You can already teach it a lot of tricks that you would also typically teach to a monkey. So with 100 million neurons, you should be able to do, I should say 100 million spiking neurons, you should be able to do really cool things. And I think 100 million is is within reach. Like that will be feasible within the next few years to put on one or a couple of chips. So you should be able to build a rat brain on a chip. Henry Markram will be very happy about that. Now, just for comparison, also why the rat brain makes sense. If you look at these big deep neural networks, the way back, AlexNet was about 600,000 neurons. If you go to the more modern and bigger deep neural networks, and you do the math, because typically people give parameter numbers, but if you map back and see how many neurons are there in that network, you're talking about a few tens of millions of neurons. So if you compare to the rat, it's not that different. And we can do this a lot of times. So as I said, real neurons spike, and I think most of you know exactly what a spiking neuron is. But just as a quick reminder, let's see, I get this going. So real neurons communicate mostly with pulses. These are your spikes. A spike travels along an axon, and then it reaches a synapse, and then some complicated stuff happens at the synapse. So when the spike arrives, the arrival of the spike causes synaptic neurotransmitters to be released. These neurotransmitters then influence the next neuron, the target neuron, and they can have different ways, different kinds of effects. Usually we think about it as having a positive or negative effect. And effectively, say that a positive spike arrives, the membrane potential of your target neuron will first increase, and then because the neuron is kind of leaky, it'll start leaking out, and the membrane potential returns to its resting potential. Now, if you have a bunch of input spikes that are all positive or excitatory, then the neuron reaches some kind of a threshold, and a spike is emitted to the next neuron, and a reset happens at the spiking neuron. That's a very basic description of the operation of spiking neurons. And from that, of course, we have developed artificial neural networks. And the first one, funny enough, was the one by McCulloch and Pitts in '43, was actually very closely modeled after a spiking neuron. So they had these input values, which they took to be binary, 0 or 1s. Then there were weights that were mapped to the synapses. So that was the strength of the connection. And then the internal value was computed as a sum over the binary inputs times the weight, over which some threshold was applied, and then a binary value goes out. Now, if you look at a short time interval to a spiking neuron, this is what you would see. Of course, then, later on, artificial neural networks evolved to become the current ones that are analog. So analog values are being sent along connections, multiplied by the weights, added to the internal state. A transfer function is applied, and you get an analog value out. And here, there's actually a very different paradigm. So here we are modeling the firing rate of these real spiking neurons. We're somehow assuming that the value that is being communicated is communicated through the number of spikes that a neuron emits. Now that's a little bit crude, and that's one of the reasons that got me into these neural networks, because a lot of biological evidence shows that the rate coding metaphor is not quite correct. At least there are a number of examples in biology where there is really undisputable evidence that the spike timing matters and not so much the rate. And I think the most famous one, of course, is the barn owl, where we know that it uses spike time differences to localize prey within its hearing field, or at least to determine the azimuth. And there are similar data for the electrical fish. And the whole idea of having precise spike times to communicate information, it's attractive because you can potentially put much more information in spike timing. But there is really much more to biological neurons than these spike times. And I thought it was worth pointing out. So because we kind of forget about it very quickly if you go into very artificial spiking neural networks, but there's really a zoo of different neurons. There are many different types of spiking neurons in the brain. And we don't always know what they do. We have maybe this notion that there are inhibitory neurons and excitatory neurons, but there are many different ones shaped in very different ways. There are also many different neurotransmitters, not just excitatory and inhibitory, and there are different excitatory and inhibitory neurotransmitters, but there are also modulatory neurotransmitters that really change the input-output function of a neuron, or that might somehow make it learn faster or not unlearn. So there's a lot of those things going on in the brain. We don't quite know what they do. We also know that these synapses are pretty complicated. They are not easy things to work with. There are a lot of complications going on that we also don't have a good handle on. For example, these neurotransmitters are released through little vesicles, little bulbs of neurotransmitters, and they're actually quantized. So if a spike arrives, a certain fixed number of like 3, 4, 5, 8 vesicles is released. And the number of released vesicles is to some degree history dependent. It's stochastic, it's dynamic. So there's a lot of dynamism going on with the synaptic weights. The other thing with synaptic weights is that especially small weights or small synapses are very transient. Neurons connect to one another, and if there is no strengthening of the connections, then very often the connections are broken again and new connections are made to different neurons, or even different locations on the neuron, because in real neurons have a spatial extension. They have spatial dimensions. That you have these dendritic trees, and it matters where the synapse connects onto the next neuron. It might just attenuate. If it's further away from the cell body, the spike might be attenuated or even elongated, have a longer delay even. There's also the suggestion that dendritic trees can integrate and threshold on their own. So you would have a multi-compartment, multi-computational unit that does useful things on it. I think actually MobileNet does something very similar to what we think quite a few pyramidal neurons do, for those who are into the deep learning literature. And the other thing is that there are these neuromodulators like dopamine and serotonin that are emitted from just relatively few neurons that have very broad projections into the brain. So dopamine, when it's released, is really released to quite large areas of the brain, and it may or may not be differentiated. There's a lot of discussion still going on in neuroscience, but it's not like there is one dopamine neuron targeting one other specific neuron. It's quite broad. It really is modulating as their effect is. Now things are also a lot more complicated at the spiking neuron level. I think everybody's seen the Hodgkin-Huxley model, which is a very careful description of the biophysics of what's going on in this axon of the squid. And it carefully models the gating dynamics for potassium and sodium channels, etc., with a bunch of variables in there that actually can be set, and you can get different behaviors out of your spiking neuron depending on those values. So Zikevich gave a really nice overview there. So by playing around with the parameters of your Hodgkin-Huxley model and within reasonable physiological ranges, you can get very different behavior out of it. And that's behavior that's also observed in real neurons. So you could have, given a fixed stimulation, you could have just simple spikes coming out at a fixed frequency, which is the sort of the ideal thing we observe, we think about when we stimulate a neuron. But very often, you first get a burst of spikes before you get a fixed discharge, and there could even be a delay, and there are all kinds of other behavior going on in these neurons. And like I said, synapses are pretty complex as well, and there's a lot of details going on there that are barely understood, let alone used in some way. And one thing I want to pick out is that real neurons have this thing called adaptation. And adaptation has to do with the fact that the input-output response curve of a neuron can actually change over time as a function of the dynamics of the signal that it receives. So here's the classical example. These are recordings from the blowfly. And the blowfly, of course, is an insect with not that many neurons. So we really can pick out these H1 neurons, as they are called. And the H1 neuron seems to code for turning velocity, torque. So you can put the fly onto this equipment. There are not that many ethical problems with working with flies. So you can put it on such equipment, and you can make it turn as you go. And you can make it turn exactly how you want it to. So you can have a specific trajectory through velocity space, and you can repeat it again and again. And what you can also do is you can, of course, double the speed. And when you double the speed, now there's an interesting thing happening. You give it the same curve, and you see that actually the same firing rate comes out after adaptation. You need to wait a little while. If you change the velocity, first the firing rates go up, and then they go down again, and they settle. And they settle all the way until you have pretty much the same firing rates, given twice the velocity everywhere. And adaptation is something that is observed in large parts of the brain. And what we think it does is that it adapts the real neuron to signal contrast rather than signal itself. There's a lot of evidence for that, and it means that perhaps computing with contrast is a much more useful quantity than computing with signal. That's at least what biology seems to suggest, and it's one of the things that I still want to try out. But adapting neurons are useful, and I'll give you an example a bit later on. But some details. Adaptation, one of the curious things about adaptation is that it follows a power law dependence. So what do I mean by that? I mean that if you inject a fixed amount of current into a neuron, what you will find is that first it fires a lot of spikes, and then it starts to fire at a lower and lower firing rate. So this is the typical spike pattern that you would get out if you get this kind of current stimulation into your neuron. And if you plot that as a firing rate, you see a firing rate decay. And if you do the math, you find that this decay actually follows a power law. So it keeps decaying and decaying and decaying until very long time scales, but then it starts to decay only a little bit. And the hypothesis is actually that this kind of spike rate adaptation does a dynamic adjustment to signal statistics over multiple timescales. And there's beautiful data from that, from like Adriana Fairhall and Cristiano Pastorini. And there's the anecdotal observation, which is kind of neat, but it might actually be important, that the way we consolidate memories follows a very similar timescale behavior. So it could be that these two are very tightly linked. So why am I mentioning the spike rate adaptation in a bit more detail? Well, remember that I talked about the neurotechnology that's taking off, and of course people are now starting to put in real visual prostheses into people, which work, at least for monkeys, it works reasonably well. It's amazing. If you the chance, go look at one of those videos. The monkeys can really see, even though they have only 1,000 pixels, they can see numbers and even outlines of objects. But a much more mature technology is the one with cochlear implants. And it's close to the same kind of neurotechnology. So a cochlear implant is implanted in patients when they have a real loss of hearing because of damage to the hair cells inside cochlea. So that's here. There are these hair cells, and they move when there is air, and they then generate spikes that go further into the hearing system. And what you try to do with a cochlear implant is, because these hair cells are damaged, you try to simulate the electrical pulses that they would have generated. So this is very, very important then to understand how electrical pulses are created in response to sounds. So that's why you have a sound processor attached to the whole system. It has a microphone, it records sound, and the sound is then transmitted as electrical stimulation into the cochlear implant. And this is a long way of explaining that what people complain about when they have cochlear implants is one very simple thing. They complain that the sound is either too loud or too soft. So that's interesting from a patient perspective, but also from a scientific perspective, because now we start to wonder, we know this effect, and then we also find that the sound processor, the way they compute the electrical impulses, is actually not taking into account this long timescale adaptation of real neurons. So as a first step, we've been able to show that, that to make a good fit for these hair cell activations, you do actually need to have a power law adaptation. There's very little data over extended time periods, but with power law adaptation, you get a much better, you get the correct curves for your spiking neurons, the hair cells. And then of course, we hope that we can demonstrate that solving this problem in your sound processor will actually give you proper perception of sounds. And so the problem with loud and soft sound should go away. And of course, since vision is not that different, people should have the same problem with vision, but we'll have to wait until implants to get people to report on that. And the same technology should help there. So there's one more thing I want to complain about, which is spike timing dependent plasticity. It is not what you think it is. So it's been a workhorse in many applications, or in many chips and algorithms. People use this data from BMPU from '97, again, a good year, where you have this kind of plasticity that they measured in a neuron, depending on the timing between an input spike and an output spike. And the famous result from BMPU was that if the presynaptic input came before the postsynaptic spike, then the synapse was potentiated, so the synapse became stronger, and that depended actually on the time difference. So the closer the time difference, the higher the potentiation of the synapse. And similarly, if the presynaptic input came after the postsynaptic target, and it was essentially acausal, then there was a depression going on. So the synapse became weaker. And that's a very nice story, and it suggests there is some kind of a good spike timing dependent learning rule that you can just use, and people have spent a lot of time and effort modeling it. But the crux is that this is a bit of a, what do you call it? This is really a snapshot of learning, and there are a lot more parameters going on. This is biology, it's complicated. But this is really the nicest picture you can make for plasticity. If you do, because some things will are just a little bit more complicated. So just to give an example, the whole potentiation, the increase of weights, is dependent on how often you are pairing, or the frequency of which you are pairing pre and post spikes. So if their frequency is high enough, you get your potentiation. But if you decrease the frequency, so the interval is lower, like once per second or once per few seconds, then the LTP actually becomes LTD. So this positive part becomes negative. So the positive part depends on the pairing frequency, and there are a lot more details. I would suggest also to go read the real paper by BMPU. It's a good paper, but you also need to read the captions. And somebody who did all the legwork and the fantastic collection of what actually goes on in biology is Claudia Klopat. And almost 10 years ago, she already published a paper that captured all these different behaviors that underlie real spike timing dependent plasticity. And then she arrived at a learning rule that's described here. It's complicated. You have to carefully read the paper again to get this. But this captures what happens in biology much more closely. Of course, that's all good, but we still have this problem. Now we have a better phenomenological description. But that doesn't really help us. We still need to think in terms, as a machine learning person, you're thinking in terms of loss functions. So what are you optimizing? What's the learning goal? And I did work on that a bit a while ago. So we showed that this STDP curve, including some of the more complications of LTD and LTP, you could model it if you posit that the neuron is minimizing conditional entropy. But to be very honest, I don't know what the loss function, what useful loss function minimizing conditional entropy is. So it does give you STDP curves that are more biological with this goal, but I'm not sure why you would use this goal. Writing up this keynote actually gave me some new ideas, so hopefully after the vacation we can make a better goal. But this needs some work. So finally, one other thing I want to comment on is the whole idea of spike rates. It's got into our artificial networks in some way. Most experimental neuroscientists will just talk about firing rates. They plot firing rates. But getting firing rates in an experimental neuron is almost as difficult as a spiking neuron has difficulty with interpreting firing rates. It needs a little bit of thinking. So how do you get a firing rate? Now, the most naive thing is to say, well, I'm just going to wait for a second and I'm going to count until how many spikes I see. Because that's literally the firing rate. But that's not what happens in a spiking neuron. There's not a little man counting spikes for a time window and then reacting. Of course, we can compute an instantaneous firing rate. We just say, well, firing rate is 1 over the time difference. So we just measure the time when we get our spikes and we do the inversion. But again, that doesn't really fit on that model of spiking neurons. One alternative is to have populations, like we use 100 or 1,000 spiking neurons that are all roughly tuned in the same way. And then at any small time window, we can just look at how many neurons are active. That works. It's a form of population coding, and it's incredibly wasteful. And there are no 1,000 H1 neurons in the blowfly. So we know that at least in insects, that's not the case. It turns out we can do this actually quite effective. It's not such a huge problem once you realize that the spiking neuron does something very similar to sigma delta coding. And I can walk you through this fairly quickly. Suppose that you have a spiking neuron that receives a bunch of spikes. And the spikes actually cause the membrane potential to follow this trajectory if there were no spiking. So the spikes come in, the signal increases and it decreases again. But of course, in reality, there's a threshold. And when the spiking neuron reaches threshold, it will fire a spike. And we can model what happens to the potential actually as subtracting a fixed kernel. This is thinking as a spike response model. So you're subtracting this fixed refractory response, and then current keeps coming in again. Oh, sorry. And then also the spike arrives at the next neuron where it will deposit its input. So this could be your postsynaptic potential at the next neuron. Of course, current keeps coming in. I subtracted the refractory response, so my value here is now below threshold, but current keeps coming in again. And at some point, I'll reach threshold again. I emit the spike, and the refractory response is subtracted again. The spike actually adds to the next neuron, and we can repeat the process. And the effect is that your spikes added up at the next neuron follow some sort of a transfer function. It's a little bit noisy. There's some shape transfer, some nonlinear mapping between this red curve and the blue curve here. But there is a mapping, and we can actually compute it. So this is your effective transfer function. It's going to be a little bit noisy because these spikes have this wobbly bit around the mean, but it does work quite nice, quite well. And other people have noticed this as well. You can actually make a very close correspondence between a spiking neuron and a sigma delta modulation encoder. Yoon in 2016 noted this, and others before that already. If you look at what a spiking neuron does, where it takes spiking input, there is a membrane filter, there is a nonlinearity spiking mechanism, effective kernel is subtracted from your input and a spike is sent to the next one. That's a very crude and quick overview of what spiking neurons does. And then if you map that to your APSTM, asynchronous pulsed sigma delta modulation, then you can get a one-on-one correspondence. So in that sense, spike rate coding with potentials works. It works quite well. We've worked with it for quite a few years. It works quite well even for fairly low firing rates. So if you do this in a naive way, you're going to get firing rates out of it of hundreds or thousands of hertz, which is a bit much. It's not going to give you much of an energy advantage in the end. But if we use different neurons, we can get to very low average firing rates, like 10 or 20 hertz, to do really useful things in the network, provided, of course, your spikes are long enough. So just to summarize the biology, real spiking neurons are much more complex than leaky integrated fire neurons, and so are synapses. We think some of this complexity is highly functional for SNNs, and definitely for neurotechnology. And popular local learning rules like STDP are mostly non-motivated phenomenological artifacts. And finally, there's a close correspondence between rate coding with spiking neurons and sigma delta ADA conversion. So at some point, I would say the story there is that nature found a way to communicate analog values over a cable in an efficient manner, basically by using pulses, and then reconverting the signal back at the synapse into an analog contribution to the next neuron. I would note that in very low firing rate regimes with short time constant, this does not work very well. So that brings us to my favorite philosopher. All models are wrong, and boy, did we get that the last couple of months. But some are useful. So I hope to show you at least something about all these biological details that help us make better spiking neural networks. And a lot of these things are still unexplored, or there are just some initial ideas. So there's more to it, and a lot of work to be done. So I go back to the original notion of energy efficiency. We would argue that if you want to get energy efficiency out of spiking neurons, you need to consider things that are going on through time. Because if you consider a standard neural network having to process video at 60 frames per second, it's really a huge job. It needs to communicate every activation value to every other neuron at every frame. And if we can somehow do this cleverly with spiking neurons, then we could do that with 1 bit per spike once in a while. And of course, then we also can look at spiking neurons, and we see that in a spiking neuron, every spike causes just the addition of a weight to our spiking neuron, whereas an analog neuron involves multiplying the activation value with a weight and then adding it to the activation step. So that's one multiplication that we have to do every time step. And that's where a lot of our potential for energy efficiency comes from. So what are continuous time problems? Looking at the time, I can only show you something about signal analysis, some of the work that Bojan did recently. I can point out some of the cool things about that. But similar applications are there in robot control and also in cognition. So we can do spike-based reinforcement learning with biologically plausible deep learning settings. It's a bit of a hassle, but it does give you monkey tasks, so monkey intelligence. So here, for this work, we focused on the idea that that for spiking neurons, the best tasks are tasks that are trying to classify streaming information. So we have a stream of information, and at every time point, we have seen some of the data, and we need to say something about what kind of class we are seeing at that time or at a particular point in time. We want to do that for every point in time. And ideally, we don't want to have a huge buffer that needs to reuse all the data all the time. And this is a slightly different problem than the one that is very often encountered in time-sensitive tasks, where basically there is some kind of a segmentation algorithm running before, and then the question that is asked is what is the class within this already segmented part, which is actually an easier task. But we're interested in this task where we get a classification out. This is in case of an ECG signal where you would want to classify which part of the wave you are in at any particular time. So we developed a spiking recurrent neural network with multiple layers of recurrently connected spiking neurons, 1 or 2 layers actually. Then we had some sort of a readout spiking neurons that didn't really spike, but otherwise they behaved as spiking neurons. And we applied error back propagation through time to this network. And of course, error back propagation through time requires that you back propagate your error through all the unrolled network size, or the unrolled network. Now, what we already known for a long time is that with spiking neurons, doing this is a little bit tricky, because as you go through your derivation chain, the chain of derivatives, you go from spikes to weights to potential to spikes. And going in the other way, how do spikes vary if I change the potential, which is the thing you're interested in? That has always been a tricky part. I know it was one of my first papers on how to do that. And there I took a very crude approximation. The spike will change in a linear fashion when you change the potential. That's of course not true, because if you lower the potential enough, it will not spike. But as an approximation, it worked then, and people like Emre Nefce and Friedemann Zenke have been developing this much further and in a much more rigorous manner in the form of surrogate gradients. And the really, really cool thing about surrogate gradients is that if you use a surrogate gradient, you can use PyTorch. And PyTorch means a lot of the hassle of building your own spiking neural network goes away, and let alone training it, and let alone doing all the math for doing error back propagation through time. So that is all taken care of by your auto differentiators. And then all of a sudden we can also do these somewhat more awkward things, like also learning the parameters inside the spiking neurons. So that's what we did. And we checked out 2 different spiking neurons for these tasks. One was a very simple live neuron. And if you look at the live neuron from a bit of an abstract point of view, then you could say that it's its own recurrent neural network, because it is self-recurrent within the membrane potential that decays. So if you tune the membrane potential parameter, you can tune the recurrent memory essentially in the live neuron to the dynamics that you need. We can also make a much more, well, a somewhat more complex neuron, like the adaptive neuron, where we don't just have the membrane potential as an internal loop, but we also have a threshold that is being changed upon every spike, and we can change the decay rate of the adaptation. So adaptation in this model increases the threshold when there is a spike, an output spike, and this adaptation then decays with a time constant. And we can change that. We can make it fast, we can make it slow, and we can train all these time constants with PyTorch. I'm explaining that because with such a framework, we can actually get really, really good results. We got what we would argue is new state of the art for spiking neural networks on tasks like the sequential MNIST and also on the permuted sequential MNIST. We also got new state of the art for the spiking Heidelberg data set that Friedemann Zenke put out just last year. So this gets you pretty good performance, although still quite far removed actually from a CNN. Where we note that our neural network is using 256 spiking neurons, and a CNN, once you do the math, it's actually 1 million neurons. And the gap is not that large if you look at it from that perspective. And finally, for the ECG application, we used only a small network, only 46 neurons, and we got state-of-the-art performance out of that. Better than LSTM or GRU or vanilla RNNs, even better than bidirectional LSTM values. And that all with reasonable firing rates. So these firing rates are percentages. So for every time stick, the probability that the neuron fires is given. So these are fairly low numbers. So why does it work so well? So we did an ablation study. The first is that it really helps to have this adaptive neuron. It really does outperform a standard live neuron. So that's what you see here. And it also helps to train the membrane potential, the parameters inside the neuron. So if you start ablating the different, the learning of the parameters, then you see performance going down quite a bit, 4 or 5%. And so that's one of the key improvements. Just by training the parameters to fit the data problem, we get good performance instead of performance. So the spiking neuron model makes a significant difference. And like I said, there are lots of different neurons. There's lots of opportunity to try different things. We also did a theoretical energy efficiency computation, where we really just went around computing the number of accumulates and multiply accumulates that each algorithm needs. We did ignore the memory. We assume that future neuromorphics will have very efficient memory access, or everything is put into resistive memory or memristors in some way, such that we don't have to worry too much about it. But then we can do a fair comparison. And we actually find that using spikes then gives us a factor of 100 over the best RNNs that we evaluated. And once you start doing the math for LSTMs, you realize how incredibly inefficient they are. So the difference with LSTMs is even much further, even though we are doing better or the same. So just to summarize, supervised learning with backpropagation through time and Surrogate gradients gives us high performance, theoretically low power SRNNs for these temporal classification tasks. The spiking neuron model really matters. It does help pick the right model or pick some properties. And you really do need to adapt the spiking neuron models to get good performance. But tools like PyTorch really make a difference there. So I would argue with Jan LeCun that at least here for these kind of tasks, we are getting very close to state of the art on practical tasks. And then he will argue back that we are still far behind state of the art in deep learning, of course, because there is no BERT GPT-3. How does it work with self-supervised learning in spiking neurons? And I would argue that here we probably need efficient local learning rules. Biologically plausible deep learning might be a way to get there. And I would also think that we need some kind of a GPU for spiking neurons. And I'm not sure that Loihi is yet the answer, but maybe in the future for the next Loihi. I will skip the cognition. We did model how you could do continuous deep learning with reinforcement learning based on basal ganglia circuits in real time or in continuous time. There are some citations there. We'll leave it up. We can turn it into a spiking neural network that works just as well. It doesn't need a lot of neurons. The trick really with cognition is into getting good sensory representations. If you have good sensory, abstract sensory representations, your cognition becomes relatively easy. So just to conclude, many of the features of biological neurons are not used yet, even though their biologists and neuroscientists will tell you that it's very relevant to how real neurons deal with signals. Synapses are very complex. There might be a lot more to it. Eligibility traces could be kept inside synapses. There is all kinds of short-term dynamics going on, and we don't really have a good handle on what it's doing in terms of learning. We would like local learning rules, of course, to run our spiking neural networks, but I would argue also that continual learning is not a solved problem. So if you put a chip out that is continually learning for its environment, you need to start worrying about stability guarantees. And if you do anything like reinforcement learning, you know that most of the time, if you keep learning, it falls apart. So there's the caveat. BPTT and SG make recurrent spiking neural networks work. Very pleased with that. It opens up a lot of, at least edge AI applications. And continuous cognition can be made to work. I did not give you details on that, but we are still far removed from large current deep learning systems. And this gives us some challenges. We are not close to large learning systems like transformers and modern self-supervised learning. And a lot of the training methods are really too slow to get there. These back propagation through time with circuit gradients is never going to work for big transformers. It's just not efficient enough and it's too slow. So the question is, do we need a spiking GPU and how do we get there? This is part of some of the proposal writing we've been doing, to be honest. And the other thing that's coming up in my mind recently is that we may still need to rethink very low firing rates. I've talked about firing rate models that work quite well. They don't really use a spike time coding mechanism. But once you get down to the biology and you talk to the biologists, they tell you things that make you think like, So for instance, cortical circuits are tunable to resonance frequencies, it turns out. And there is no reason why those resonance frequencies should be tuned if you really are operating only on firing rates. I find that very interesting and maybe also very cool for future research. And finally, there's been a lot of people involved in the research. I didn't mention all of them, so a big thanks to a big group of people. Bojan's been online, Federico, and some Davide Zambrano did a lot of work, and Peter Roelsman and Hjalte Rombaerts worked a lot of the biological plausible stuff. So that was all from my end. Thank you for listening.
Thank you so much. That was awesome. So we have a couple of questions in the chat. Andrew, I don't know if you want to unmute and ask your question or I can read it.
Let me see how I get there. Chat.
I'll just go ahead and read it. So what hardware have you run backpropagation through time on?
None. A standard computer. Yeah.
Okay,
I understand that running these BPTT on hardware is very, very difficult. But that's from talking to other people who are trying to do that.
Thanks.
And then the next question is significant adaptivity observed more strongly in certain neurons? If so, which kinds?
Yes, there are, it's only a subset of real neurons that show adaptation. We don't know why. It's mainly in the sensory cortices that you see this adaptation, sensory neurons. A lot less deeper, but you should also realize that deeper areas in the brain don't fire all that much. So it would be hard to observe adaptation.
Okay. So I have a question. I completely agree that we need a GPU-like system for spiking neural networks in general. And I think the neuromorphic community is the right one to look at that. So what characteristics are you looking for in a hardware system? If not Loihi, what are the characteristics that you're looking for in that sort of system?
Yeah, so we need pretty programmable spiking neurons, because I don't think people have converged onto a specific spiking neuron that will do the trick. This is not easy, because once you start changing time constants, especially if you think about analog hardware, you're inviting nightmares, I think. And the other thing is that if you are going to accelerate learning, because I think that's the big bottleneck, then you need learning algorithms that are effective and efficient, like arrow backprop, that work on chips. And I know, so Guillaume Bellec has published this EPROP algorithm, which seems very promising. So that kind of promises to be effective. But I also know that for that, you get a 3 or 4 factor learning rule that's very hard to implement on current Loihi. So you would need to do that, you need traces on your synapses. And to compute those traces times the error is probably the current challenge. So you need EPROP-like algorithms, and you need to be able to do that efficiently on a chip.
All right, awesome. That's a challenge for all the hardware people in the audience. Does anybody else have any other questions? Seeing any in the chat, feel free to raise your hand or unmute and just ask. Okay, so we have one. Have you looked at complex synapse models like metaplastic synapses? Adding to that, in terms of local learning rules, how can we address the credit assignment problem?
Those are good questions. I have not looked into metaplastic synapses, but there is fascinating data on what real synapses do. So that is definitely something that is very, very cool, and that might get you really cool outputs. So then the second problem, in terms of local learning rules, can we address the credit assignment problem? Well, there are basically just a few approaches out there. There is a version of, so I think Blake Richards wrote a very nice paper in Nature Neuroscience last year, and there was a paper from, I think, Jeff Hinton in Nature this year, on the different ways of solving the credit assignment problem in the brain. And there are 3 approaches. One is target propagation and equilibrium propagation, 1 and 2. So there you somehow clamp the output to the desired values, and you make sure that your dynamics in the network are such that you can then learn from it. And the approach that we've been pursuing here is that we show, and I kind of flashed the image by, that if you have a feedback network, you can learn most of the tasks by using cleverly formulated feedback. And then the real trick, and that's our argument, is that it's not so much about the credit assignment per se, it's about the learning paradigm. Once you realize that nature is mostly doing reinforcement learning, and reinforcement learning is about taking one action at a time, then suddenly your credit assignment problem becomes quite a bit easier. So rather than having a vector error that you need to optimize, you're now optimizing a scalar. And we suppose that the dopamine signal is actually broadcasting the error scalar all over the network. And then you can show that activity propagating back is doing the trick. So you can get an arrow back propagation algorithm for reinforcement learning which does the correct credit assignment. But yeah, this is part of the argument. Like if you want to have fast chips that do really fast learning, spiking neural network learning for you, you need these kind of algorithms based on spikes. Yeah. So I hope that answers the question. It's a fascinating topic and there's a lot of things going on.
Malika, did you have a question? Yeah.
I just had a comment more than a question. So I just wanted to say that, you know, since the community is kind of going towards this 3-factor learning rules, and as you mentioned, like EPROP and so on, I think that there is a good place for analog hardware, just to kind of defend the analog side of the neuromorphic chips. So I think that it is much more natural to implement this kind of eligibility traces with long time constant with the analog hardware than with digital. So that was one.
Yeah, that's exciting. Yeah, I mean, this is, if you think about all those traces and all the memory it's going to require, that
right, exactly.
Yeah, you might want to go there.
So we have a couple more questions in the chat. So in biology, you have a combination of spiking and non-spiking neurons. Is this a must-have or a nice-to-have characteristic for computing?
That's an excellent question. Yeah. So it's kind of like what I tried to convey really quickly with the ADDA story. I don't think it's the whole story, but if you look at where there are non-spiking neurons, they are in locations where the neurons that need to communicate are close together. So parts of the retina are non-spiking, and then this is also what you find in other animals. Then those neurons are electronically coupled, and they will influence each other without using spikes. But of course, as soon as you want to communicate with a lot of different elements, you can no longer use that spatial arrangement in the brain or in a brain. So then you need to communicate over distances, and as any engineer will tell you, if you're trying to communicate over cables, you really want to do that with binary spikes, because otherwise things get very, very complicated. So that will be my biological point of view. Like, if you want to do this, it makes a lot of sense to go for this rate coding. So at that point, it becomes a nice to have feature to have spikes, but very nice to have.
All right, and our last question, what do you think of the transformer design in ANN area that does not need LSTM but reaches state-of-the-art on NLP tasks? How might RSNNs beat those kinds of designs?
Well, the first thing we need is to be able to compute with really big spiking neural networks. Because otherwise, we're not going to beat transformers with something that contains 1,000 or even 100,000 neurons. So I think that will be the first thing. But yeah, I'm not much into transformers because they are so big, and I don't have the resources to compute them. So yeah, something to think about in a national lab, for instance.
Yes, yeah. All right, well, thank you very much for the talk. It was great, and thank you for all the Q&A.